All right, welcome back. The last lesson was pretty long, so this one will hopefully be shorter. Today we're going to talk about active transport. Last lesson was about the cell membrane structure and then passive transport. Active transport always requires the cell membrane because you need to have a concentration gradient to move against. Active transport always requires energy. The cell needs to spend energy to move stuff. Sometimes I refer to ATP as kind of like the cell's currency. It spends energy to move molecules. There are a couple reasons why the cell might want to do this. Either the cell wants to move something that's really good against its concentration gradient into the cell, or the cell wants to move something that's really bad, like a poisonous waste product, out of the cell. Substances are often transported from an area of low concentration to an area of high concentration, which means that it is moving against their concentration gradients, against the concentration gradient. Just to review what a concentration gradient is, imagine this is our people. We've got a bunch of people mingling around on one side of the room. And this is our semi-permeable membrane that people can cross. We've got more people on one side, or a higher concentration, and less people on the other side. There is a gradient because there is a difference between these two concentrations. People will naturally move with the gradient, meaning that they will move from high to low until the overall concentration of people is the same on either side of our membrane. This is equilibrium. People are balanced. Going against the concentration gradient would be moving people from this side to the other side, and that is going to take some form of energy. That's not easy. That's what we're talking about now with active transport. All right. There are a few things here that you don't need to know about this diagram. I don't care if you know what a periplasmic binding protein is. But here we have a transport protein, and we're going to say that on top here, this is our extracellular space. So this is outside the cell. And down at the bottom here is our cytoplasm. That's been labeled for us, but that's the inside of the cell. Our desired solute is these red little, or these pink molecules. This is our, we're just going to say this is our, oh, it says here our substance to be transported. Don't need to write that there. Note that we only have 5 of these outside of the cell, which means out here we have a low concentration. And inside the cell, we have a high concentration. We are going to be moving the particles into the cell, which means the particles are moving against their concentration gradient. Naturally, they want to move from this area of higher concentration to the area of lower concentration. In order to move them inside the cell, we have to spend ATP. This is our energy. I'm going to highlight the ATP here. We have to spend energy to move against the gradient. Again, your cell's going to do this if it really wants to keep something inside or if it really wants to keep something out. Here's a membrane transport simulation here. Let's see if I can zoom in a little bit. Okay. Here's our extracellular space on the top. Here is our cytoplasm, our intracellular space. we're going to add a whole bunch of molecules, blue molecules, to either side. We're going to add a bunch to, I will just add them to the outside. You'll see that these blue molecules readily diffuse across the membrane. They're moving both ways across the membrane, but pretty soon here we're going to reach an equilibrium. Because they all started on the outside, they moved this way, but now we're at equilibrium where we have around 20 of these molecules on either side of the membrane. Let's say that this is a nutrient molecule, and we really want more of the nutrient inside the membrane than outside. We can add a transporter. These are called uniporters. It's a type of active transport. Uni means one. They're only going to go one direction. These uniporters keep the blue molecules inside, but they cost energy in order to use. This is going to mean that overall, more blue molecules should be moving into the intracellular space than in the extracellular space. And so we'll just add a few more blue molecules to play with in the extracellular space here. But as we continue to keep transporting these blue molecules into the membrane, we're fighting against diffusion here, but we should see that the concentration in the intracellular space is going to be higher. Now, we don't see that it's making a huge difference here. You need a lot of uniporters to combat diffusion, but you're basically forcing more of these to go to the inside, to the intracellular space. So the overall equilibrium will have more on the inside than the outside. Your notes here say that there's two types of active transport. That's not completely true. There's more than two types of active transport. These are two additional types. This is a transport protein that requires energy. That's a type of active transport. Here's two additional types: endocytosis and exocytosis. Endo means within. Exo means outer. Within cell process, outer cell process. Endocytosis is using the membrane and energy in the form of ATP to bring materials into a cell. Exocytosis is the opposite, using the membrane, and once again, energy is required here. It brings materials out of the cell. Exo, think exit. Exiting the cell. Here's a diagram. We have materials outside of the cell. They're bundled up in a vesicle here that goes and joins the cell membrane, and the cell membrane grabs it, forms around it, and brings it into the cell. The opposite could also be true. We have a whole bunch of materials that we want to get rid of. We package them into a vesicle, and then that vesicle fuses with the membrane, and we expel these out of the cell. Exocytosis, these exit the cell. Both of these processes require energy, and they're used to move against a concentration gradient. Cellular transport is important for a lot of different medicine. Here's an example of how you can use the principles of osmosis to help treat wounds. Indigenous people treated injuries and wounds, like a cut or a burn, with honey to aid in the healing process. Honey is high in solutes, which means it has a low water concentration. Remember from the last lesson that if you have a high solute concentration, you have a low water concentration. This means that the water from the wounds moves from the skin, because that would have an area of higher concentration, to the honey, which is the area of lower water concentration. This will dry out the wound. This dries out the wound and kills bacteria that could infect the wound. The honey also works as a barrier. It's a thick liquid, and it will work as a barrier to keep pathogens, which are organisms that might try and make you sick, out. So it dries out the wound and it keeps pathogens out. Many viruses have specific parts that fit a specific shape of a protein on the surface of the cell membrane, like a receptor protein. Scientists are trying to fight diseases by blocking a virus from entering the cell. You could use a medicine to block the receptor protein to prevent the virus from trying to go in. So imagine if the virus had a key to get into the house, but then you cover up the lock with concrete with your pharmaceutical here to prevent the virus from attaching. Similarly, many pharmaceutical drugs work by binding to membrane proteins. So for example, a painkiller drug can bind to a receptor protein to block the pain signal. That would be an example. Or an antibiotic might bind to a receptor or to an enzyme on a bacteria's cell membrane. Last thing to talk about in this topic, surface area and volume of cells. The question asks, why are cells so small? The reason cells are so small is to maximize the ratio between surface area and volume, which will allow exchanging nutrients as efficiently as possible. Let me give you an example of why this is the case. This is my cell. The surface area of the cell is the area on the outside of the cell. The volume is the space inside the cell. Particles move into the cell through the cell membrane. Watch them diffuse through our cell membrane. We want a big surface area so that lots of nutrients have spaces to move through the cell membrane. Water also moves into the cell through the cell membrane. So water is moving into the cell. Notice that if the volume is smaller, the cell fills with water faster. A large volume means it takes a longer time for the water to reach the center. You want a small volume so that there is less distance to travel. If the volume is too big, then you end up with too much distance for solutes and for water to travel, and this can kill our cells because they cannot efficiently move nutrients in or move wastes out. Picture a cubic cell. As the cell grows, what happens to the ratio of surface area to volume? Let's try calculating the surface area to volume ratio of different sized cubes. Let's say that the side length of our cube is 1 centimeter. This is our cube right here. This is our 1 centimeter cube. This over here is our largest cube, our 4 centimeter cube. The surface area formula for a cube is 6 times the side length squared. So our surface area here is going to be 6 times 1 centimeter squared, which will get us 6 centimeters squared is our surface area. Our volume is going to be the side length cubed. So that's going to be 1.0 centimeters cubed just gives us 1.0 centimeters cubed. Square units are our units for surface area, and cubic units are our units for volume. The ratio between our surface area and our volume is going to be a 6 to 1 ratio. A 6 to 1 ratio. If we increased the side length of the cube to 3 centimeters, then our surface area would be 6 times 3 centimeters squared. So remember your order of operations here. We'll do the square first, so that will be 6 times 9 centimeters squared, which will be 54 centimeters squared. Our volume is going to be 3 centimeters cubed. 3 cubed will give us 27 centimeters cubed. Now the ratio now between surface area to volume will be 54 to 27. We can simplify this ratio. Both of these things are actually divisible by 27. This is a 2 to 1 ratio of surface area to volume. If we make our cell bigger to 4 centimeters, then 6 times 4 centimeters squared is going to give us 96 centimeters squared as our surface area, and 4 centimeters cubed will give us 64 centimeters cubed. This is a 96 to 64 ratio, which in simplest terms would be a 3 to 2 ratio. You don't need to do that much math for this unit in Science 10. What I want you to take away from this is that as we increase the side length of a cube, the volume grows faster than the surface area, which means that our surface area to volume ratio decreases. As side length increases, volume grows faster than surface area. So if we want to have the most surface area possible and the least amount of volume, then we need the smallest side length, because cells require a high surface area to volume ratio, which means they need a small side length. To survive, they need a large amount of surface area exposed to the environment with which to exchange nutrients and wastes. If very little surface area is available, the opportunity for taking in materials and getting rid of materials is limited. It will be less efficient. And if a cell is unable to take in enough nutrients or get rid of enough wastes, it will die. So smaller cells are more efficient than larger cells. Because cells cannot get very big, in order to have larger organisms, they are made of many cells. So the cells themselves don't just get bigger, you get many cells, and then they specialize for certain functions. So multicellular organisms will have many types of cells in order to try and combat the problems with surface area and volume. That's going to be it for this lesson. There's some good review here. And then in the next lesson, we will start to talk about plants.